Good afternoon, class and demonstrator. My group and I decided that we would review the article Diagnosis of Thalassemia using fluorescence spectroscopy, autoanalyzer, and hemoglobin electrophoresis, which was written by Sandhanasamy, Fatma, Muhammad, Karapaya, and Vadivel. Thalassemia is a blood disorder which is characterized by the imbalanced synthesis of globin chains. The world's population currently has a prevalence of 5% with hemoglobin disorders. Thalassemia is an inherited blood disorder which is characterized by less oxygen carrying proteins, also known as hemoglobin, and fewer red blood cells in the body than normal. The hemoglobin molecule in a normal adult has four subunits, usually 2 alpha and 2 beta. However, patients with thalassemia do not produce sufficient hemoglobin alpha because of mutations in the genes that code for the globin proteins, thereby disrupting the generation of normal alpha and beta polypeptide chains. Thalassemia occurs when one or more of these subunits are faulty or deleted, and it affects the hemoglobin's molecule structure, which also affects their function, which in this case is the ability to carry oxygen in the blood to the cells. Many methods are currently used to diagnose thalassemia. The clinical history and hematologic evaluation are observed via a complete blood count that is then analyzed via an automated analyzer, which provides a better read of the various blood components for both sexes in all age groups. In addition, red blood cell morphology and reticulocyte counts help differentiate patients with thalassemia from those with other hematologic conditions. Electrophoresis is the most common method for hemoglobin evaluation, as different hemoglobin molecules such as HbA, HbA2, and HbS have different charges, which make them move at different speeds in the gel via an electric field. Abnormalities in the Hb proteins alter their charges, which can be identified by changes in their speed of migration in the gel. Recently, cation exchange high performance liquid chromatography has become a popular method for the detection of hemoglobin subtypes, as well as the polymerase chain reaction, which can be used for the detection of gene deletions in thalassemia. Spectral diagnosis using fluorescent spectroscopy is based on interaction of photons with fluorescent blood biomolecules, which is a newly evolving technique for the detection, diagnosis, and monitoring of diseases such as different types of cancers, sickle cell anemia, and of course, thalassemia. There are two main aims in this study. The study aims to compare the performance of fluorescent spectroscopy, which is a very simple and inexpensive method, with the old conventional techniques for diagnosing thalassemia, where the red blood cell counts and levels of Hb, HbA, HbA2, and HbS are estimated via conventional methods of complete blood count and hemoglobin electrophoresis to diagnose thalassemia. This experiment was also performed to evaluate and compare the performance of fluorescence spectroscopy with those of standard modalities. For example, as mentioned previously, complete blood cell count using an automated hematology analyzer and electrophoresis in the diagnosis of thalassemia.
And now onto our materials control and method. There were 50 blood samples collected from each group, the control group and patients with thalassemia trait and disease. Of the control group, there were 30 men and 20 women without a history of thalassemia or any blood-related diseases. And as for our methods, as mentioned by Jason, an autoanalyzer is used with 5 milliliters of blood collected in EDTA-coded tubes for all patients, and then recording the red blood cell distribution and complete blood count parameters. And then we have hemoglobin electrophoresis, which uses hemolyzate, which were prepared by adding blood to hemolyzing reagent, mixing and incubating at room temperature. These concentrations were recorded at 0.25 grams per deciliter, and more blood was run for samples. The third method is spectrophlorimetric analysis, which in this experiment is being compared to electrophoresis and complete blood count diagnosis. This is performed through fluorescence spectroscopy, which uses a beam of light to excite electrons in the molecules of compounds to cause them to emit photons. This is then detected for measurement and identification for changes that occur in the molecule due to energy changes and frequencies. This was performed on the blood plasma samples and the red blood cells of patients with thalassemia. In our results section, the first figure shows synchronous excitation spectrum of the controls and patients with thalassemia. This is the figure on the left-hand side of the screen. The five main peaks in order from left to right of the graph is circled in red. which represent tyrosine, tryptophan, NADH, FAD, and bile pigments. Compared to the controls, the relative intensity spectrum of samples with thalassemia was lower with samples below 370 nanometers, but higher at 450 and 505 nanometers. Using figure 1, R1 and R2 and R3 values were gathered and quantified by the intensity of the diffraction peak profile of the sample groups taken, using a reference intensity ratio to formulate a ratio to scale the control and thalassemia group to a common reference point. As you can see in the table on the right hand side, both R1 and R2 values of the thalassemia sample were higher compared to the controls, with the higher values in thalassemia disease as compared to those with the trait and the controls, which is indicative of excessive hemolysis in thalassemia disease. In figure 3 we have the synchronous emission spectra of sample groups which show the relative intensities at 325 and 363 nanometers, which were lower in the thalassemia trait and disease samples than it was in the control, while the peak at 525 nanometer was higher in the thalassemia trait samples when compared to the other two. In figure 4, the R4 and R5 values were derived from the SES graph in figure 3, showing that there were two distinctive differences observed in the values, as they were higher for thalassemia samples when compared to those of the control samples. In figure 5, we have the fluorescence emission spectra, which shows the spectrum of acetone cellular components, with the peak of control at 475 nanometer, circled in red, corresponding to the Raman band of acetone, in which biomolecules are suspended, while the other two major peaks at 585 and 635 nanometer represent the different forms of porphyrin in its basic and neutral form. And we can see from the curve that the thalassemia curves are lower, which is tabulated into a table which shows that the control is higher than the thalassemia trait and disease. In figure 6 we have the conventional methods of complete blood count and hemoglobin count to identify the thalassemia through the lower red blood cell and hemoglobin levels. Patients with thalassemia present with low counts in both indices, which are seen in the sample groups identified with thalassemia, and is significantly lower than the controls. And our last result, figure 7, shows hemoglobin electrophoresis in all sample groups. Overall, compared to the controls, the hemoglobin A values were lower in patients with thalassemia, whereas the hemoglobin A2 values were high in patients with thalassemia than the controls. The results show that elevated levels of hemoglobin A2 is seen in beta thalassemia patients, with also reduced hemoglobin A values due to compensation of lack of beta globins. Overall, the experiment shows that there's an 85% accuracy between conventional method of complete blood count and electrophoresis of hemoglobin versus fluorescence microscopy, which demonstrates its significance while also being an inexpensive process to use.
This prospective study aimed to evaluate and compare the performance of fluorescence spectroscopy with the traditional methods like automated hematological analyzer and hemoglobin electrophoresis to diagnose thalassemia by analyzing 50 blood samples from each normal and diseased subject. First, the conventional method like complete blood count and hemoglobin electrophoresis were used to confirm the diagnosis of thalassemia among the diseased population. Then the fluorescence spectroscopy was used to analyze the level of blood biomolecules like tyrosine, tryptophan, NADH, FAD, and bile pigment. Both the relative intensity results from synchronous excitation spectrum and synchronous emission spectrum were considered when analyzing the results by spectrophotometric analysis, as shown earlier on the results section of the slide as well. So the five main peaks at 275 nanometer, 290 nanometer, 370 nanometer, 450 nanometer, and 505 nanometer represented amino acid like tyrosine and tryptophan, coenzymes that are NADH and FAD, and the bile pigment respectively. All the spectral findings were where the relative intensities value were lower at below 450 nanometer and they were higher at 450 nanometer and above for the thalassemia trait TT and thalassemia disease TD samples compared to the control samples, and all these results were consistent with the previous studies as indicated on the brackets here. So the detailed reference about these studies will be listed on the reference section at the end of the presentation. And these studies indicated that all those amino acids and the coenzyme biomarkers are engaged in several cellular redox activities. The previous study by Abdul Razak and group found that the tyrosine level in patients with thalassemia were lower compared to the normal samples, also shown on the results section of this presentation, which also supports the findings of this study. Similarly, the low levels of tryptophan in TT and TD samples is also backed by other studies, which shows that the low production and excessive damage of tryptophan leads to various types of anemias, including thalassemia. Moreover, studies suggested that the reduction in the enzymes NADH and FAD results in lower production of red blood cells, causing anemia and other abnormalities, as shown by the study done by Kuhn and Group 2017. We also observed the faster rate of red blood cell decay in patients with thalassemia in comparison to normal people, which is most likely due to the very high levels of porphyrin and excessive amount of abnormal red blood cell production, as shown by previous studies done by Stockman et al. On the other hand, while analyzing the conventional technique results, the parameters like red blood cell count, hemoglobin, and mean cell hemoglobin were lower in samples with thalassemia trait TT and thalassemia disease TD compared to the normal group, which is also supported by earlier studies done by Pengun et al., which is due to the high oxidative stress in red blood cell that also increases the rate of decay in thalassemia, resulting short supply of oxygen. Furthermore, hemoglobin electrophoresis is a useful tool in diagnosing thalassemia and sickle cell disease that analyzes the hemoglobin subtypes like HbA, HbA2, and HbS. This study had the average HbA value of 98.5% in normal samples, whereas 94.8% in TT and 78.6% in TD samples, whereas the mean hemoglobin A2 was 2.6 for normal sample, whereas 3.2 and 6.8 in TT and TD samples respectively. These results are consistent with previous studies where HbA was lower in thalassemia patient, whereas HbA2 was double in thalassemia patient compared to the normal samples. And the studies that also backs this result were done by Galanello, Ilsaid et al. And Young et al.